objective to determine the effect of light distance on the rate of photosynthesis. Introduction. Photosynthesis is a succession of complex reactions catalyzed by a variety of enzymes. To facilitate comprehension, photosynthesis can be considered in two stages. The initial phase is known as light-dependent reaction, since light is required for their occurrence. Chlorophyll absorbs light energy, which is then used to generate ATP in the process known as photolysis. The energy is also used to split water molecules into hydrogen and oxygen. The release of hydrogen ions, electrons from the hydrogen components of water and oxygen. Oxygen is a byproduct of photosynthesis, yet its release into the atmosphere is essential for the survival of aerobic species. In light-independent process, ATP, hydrogen ions, and electrons are employed. In the light-independent processes of the second stage of photosynthesis, ATP, hydrogen ions, and electrons are required. During the light-independent processes, carbon dioxide is mixed with hydrogen and ATP to produce a variety of organic compounds for the plant. Carbon fixation refers to the transformation of inorganic carbon dioxide into organic compounds such as glucose. ATP is the source of energy for the process. The reactions that occur during photosynthesis are summarized as follows. Introduction. The process of photosynthesis shows how photosynthetic organisms store light energy, water, and carbon dioxide to create glucose and oxygen. Oxygen is released in the atmosphere, whereas glucose is used at the later time to supply the energy needs of the cell. Combining of these elements to produce sugar and oxygen takes place in the chloroplast, which includes chlorophyll that associate with the actions of the green pigment. A pigment is any substance that absorbs light. The wavelength of the light reflected or absorbed determines each color of the pigment. Chlorophyll, which is a green color pigment, tends to absorb all wavelengths of visible light except green. Since chlorophyll do not absorb the light with the specific wavelength of color green, it reflects back to be detected by our eyes. This is the reason why leaves are green. The color green could not be absorbed by the chlorophylls in the cells that the light reflects back to our eyes, making it visible for humans to see. Hypothesis. If the distance from the light increases, the rate of photosynthesis decreases due to the light intensity. Material. First, I choose leaf bougainvillea, string, puncture, measurement tapes, light, beaker 500 ml to 1000 ml, filled with water, soda bicarbonate, glass, and time measurement. First of all, I choose bougainvillea plant. Using a hole punch to get 10 leaf teas from the plant that I choose. After that, put a water from the beaker into the glass, 50 ml water. And then, using a small spoon to put a soda bicarbonate 0.5 grams into the glass, mixed with it. After that, remove the plugger from the string and remove the cover from the tip. Put the leaf teas into the barrel of the string and tap them down to the tip. Replace the plugger into the string, being careful not to damage the leaf teas. Draw about 5 to 10 ml of bicarbonate solution into the string. The leaf teas should float in the solution. Hold the string with the tip up and expel the air by gently pushing on the plugger and shake it. Plug the tip of the string tightly with your finger and gently pull on the plugger, creating a slight vacuum. While you pull the plugger, must be sure that shake to make sure the air bubble comes out. You should see tiny bubbles coming out of the leaf disc. Hold the vacuum for a few seconds and then release the plugger, letting it snap back. Some of the discs should begin to sink. Repeat the previous step several times until all of the discs have sunk to the bottom of the solution. We need to tap on the plugger to release the bubbles in order to make all the leaf discs sink. When all the leaf discs have settled to the bottom of the solution, carefully remove the plugger and pour the disc and solution into the cup. They should settle to the bottom of the cup. If any leaf discs float, remove them from the beaker. Set up table lamp so that it is suspended about 3 cm above the beaker. Turn on the light, start a timer and watch the leaf disc at the bottom of the cup. Notice any tiny bubbles forming around the edges and bottoms of the disc. After several minutes, the disc should begin floating to the top of the solution. Record the number of floating discs every minute until all the discs are floating. Once finished, remove the light and put the cup in the dark for 15 minutes or cover with aluminium foil. But I'm using a box to cover the leaf. After that, repeat step 3 to 15 using the same leaf, but this time set up the table lamp so that it is suspended about 10 cm above the beaker. That's all. Discussion. First, disc of leaf will be placed in a water-filled string to suck out the oxygen in the leaf disc by drawing back the plugger to create vacuum. This is because leaves have air in the spaces between cells, which help them collect CO2 gas from their environment to use in the photosynthesis. When you apply a gentle vacuum to the leaf disc in a solution, this air is forced out and replaced with a solution, causing the leaf to sink. Second, the leaf disc cannot do the process of photosynthesis because of the soda bicarbonate serves as a source of carbon dioxide for photosynthesis. Third, the leaf disc rise to the surface of the solution because of the subsequent carbon dioxide which photosynthesis in the presence of light and leads to the accumulation of oxygen in the air spaces of the spongy mesophyll, which increase the buoyancy of the leaf disc. Fourth, the leaf disc not floating on the surface of the solution because without the light energy, no photosynthesis occur and no oxygen gas produced. Conclusion: the experiment showed that the distance from the light increased, the rate of photosynthesis decreased due to the light intensity. In the light, the experiment expects to see the disc in the solution, baking soda, should begin to rise as they use the CO2 to undergo photosynthesis and produce oxygen bubbles. The bubbles should cause the disc to float. Based on the result, the distance of light 3 cm took very quick to float than the distance of light 10 cm. After remove the light and place the cups in the dark, the treatment disc should stop undergoing photosynthesis and the disc should begin to sink.